Hi, my name is Nathaniel. As a scientist at MIT, I study how ultraviolet light damages the proteins in the human lens. And for my experiments, I purify human lens proteins from bacterial E. coli cells designed specifically to express human proteins. In this demonstration video, I'm going to show you how biochemists use column chromatography to purify a variety of proteins. In specific, today we'll be purifying GFP, green fluorescent protein. In lecture, Professor Landers talked about the incredible diversity of structure and function that proteins have. Protein purification is a critical step to understand that structure and function. By the end of this video, you'll be able to describe how biochemists use column chromatography to purify any number of proteins. Before we get started, let's talk about what we'll be doing today. How do we purify one specific protein from all of the other proteins in the cell? As Professor Lander discussed, scientists start by breaking open or lysing cells to release all of the proteins. Once they have this mixture of proteins, how do they isolate one specific protein from all of the others? Scientists can use a technique called column chromatography to take advantage of the different properties of their specific protein of interest, like size and charge. Some proteins are larger than others. Some proteins are more hydrophilic or hydrophobic than others. We can separate proteins by flowing a protein mixture through a matrix of beads in a column. The scientists select the beads for the column and the binding buffer based on the properties of the protein of interest. The protein of interest binds to the beads based on its affinity for the beads in the column. To remove other unwanted bound proteins that are more weakly associated with the column, scientists can wash the column with a different buffer that pushes the weakly bound proteins off. When removing the protein from the column, we use a third buffer and collect fractions of the liquid coming off the column. For most proteins, scientists don't know which fraction contains the protein of interest. They have to test each fraction for a specific activity. For today, we'll take advantage of the unique fluorescent property of GFP and use its green glow to track the protein throughout our purification steps. Did you know that GFP originally came from jellyfish? GFP is a really cool protein because it fluoresces green and serves as a valuable tool for scientists. Scientists have found that the protein has a beta barrel structure, a barrel shape made up of beta strands that you heard about from Professor Lander. Scientists copied the gene encoding GFP and placed it into the bacteria E. coli. So where do we produce this GFP for purification? We need to make many copies of the protein to efficiently isolate the protein of interest. Let's get started. I have E. coli cells that are designed to express GFP. They grew and divided overnight. I also have cells that don't express GFP. We'll collect these cells too as a comparison. Because it's not ideal to isolate GFP from jellyfish in the lab, we can make many copies of GFP by expressing the protein in the bacterium E. coli. How do we get bacteria to make protein from a jellyfish? Scientists copied the gene encoding GFP and placed it in the bacterium E. coli. You'll learn more about this process of cloning later in the course, as well as how scientists use GFP in research. We spin the cells at a high speed in these tubes so that the cells go to the bottom as a pellet. How do we know that our bacterial cells really are expressing GFP? We can use this really cool property of GFP that it fluoresces. I have this UV lamp here, and when we shine the UV light on the cells that are expressing GFP, they should glow green. Let's test our cells. I'm removing the liquid from the tube and saving the cell pellets. Let's shine the light on the cells that are not supposed to express GFP. What do you expect to see? Nothing, right? No green fluorescence. Let's shine the light on the cells that are supposed to express GFP. Check it out. Green glow. So how will we lyse our E. coli cells now to get at that mixture of proteins within, including GFP? I'm resuspending the cell pellets in a buffer by pipetting up and down repeatedly. Now I'm adding an enzyme that breaks down cell walls called lysozyme. Next, I'll freeze then thaw the cells quickly. This lyses the cells, releasing all the proteins, including GFP, from the cells. Afterward, I'll centrifuge the samples again to separate insoluble cell debris from the soluble proteins. So how will we separate GFP from all of the other soluble proteins in the bacterial cell? Now that I have the cell debris separated from the soluble proteins, I'll use a small column packed with tiny beads to separate the proteins by a specific property. In this case, I'm using an affinity column that contains hydrophobic beads. GFP contains many hydrophobic amino acids. In a high salt buffer, the hydrophobic parts of GFP move to the exterior of the protein, and GFP binds to the beads as the protein enters the column. The other bacterial proteins with fewer hydrophobic amino acids flow past the beads and through the column. Let's prepare our column for purification. I'm adding a buffer with the same salt concentration as our protein solution to equilibrate the beads. I'm removing the cap at the bottom of the column to allow the buffer to flow through. With that done, I recap the bottom of the column. Now I'm adding my cell extract of soluble proteins in a high salt buffer to the top of the column. Using this UV lamp, I should be able to detect any GFP. Take a peek. Glowing green. So we can see a ring of green fluorescence at the top of the column, showing that GFP has bound to the top of the column. So how will we make sure that all the other bacterial proteins are removed from the column? I'm adding a wash buffer with a medium salt concentration to our column. Any proteins with less hydrophobic exteriors than GFP did not bind as tightly to the hydrophobic beads and will flow out of the column during washing. The hydrophobic exterior of GFP has a higher affinity for the beads and remains bound. So now that all the bacterial proteins are washed away, how do we remove GFP from the beads on the column? Remember, the GFP bound the beads in the column because the beads in the GFP exterior were currently both hydrophobic. I can remove GFP from the beads by adding a buffer with low salt concentration. So what do you think the low salt concentration solution will do to GFP? The low salt concentration will bring the hydrophilic residues of GFP to the surface, so the protein will no longer bind the hydrophobic beads on the column. I'm collecting fractions of what comes off the column in several tubes. Let's see if the GFP is moving through the column now with our UV light. Yes, I can see the GFP moving through the column. Which tube contains the GFP protein? I do not know by only looking at the liquid collected off the column. For most proteins, we would have to test each fraction with an activity assay or another protein detection assay. But remember, we're purifying a protein with the unique ability to fluoresce. Let's check which fraction I collected contains the GFP using the UV light again. I can see that most of the green fluorescence is in tube 4, so GFP eluted in fraction 4. I've now purified GFP from most of the other bacterial proteins, and you've learned something about biochemistry and how we purify proteins. I hope you had fun.